Okay, let's have a class. We have some cool stuff today, so let's have a class. First of all, we have 3 patients' results. Are those all 3 patients' results for the urine sample testing? Okay, you have some. Let me talk about something here, then I'll go to your stuff, okay? First of all, we talk about UTI. So, let's look at your McConkey agar. What looks like? Is that positive? Yeah, looks like. My stuff looks like positive. It's been a while, but there is a colony there, and it's pink color. So McConkey agar, what we find is pink colony, which indicate lactose fermentation positive. So possibly it's E. coli. Look at your blood agar. What do they look like? Your blood agar, I didn't see beta hemolytic, but I do see lots of the colony growing there. Okay, so I will say positive, gamma hemolytic. Now how about your nutrient agar? So let's see what you have. This one, that's what I get. You can count how many colony there. I would say this one, if you go ahead to count, it's going to be more than 100. You can count your own. So if you nutrient agar more than 100, that means more than 100,000 CFU per ml. This diagnosed as UTI, urinary tract infection. So that is some sample from my bench. It looks like that. I have a second one, is the same thing. Positive for colony on the McConkey agar. Blood agar looks like some beta hemolytic. And this one you have lots of colonies there, but you have to count. It's the same thing for the last one. Let's look at your patient, what it looks like. Patient number 1. So I don't know who is this, just patient number 1. So very surprisingly, we didn't find anything on the McConkey agar. So that means it's not E. coli. And you have something from blood agar, and you have a limited colony, I will say roughly about like 30-ish, 20, 30-ish.
It was 38.
Yes, 38. So is this guy UTI? So let's say here you have a negative. This is your positive. So now hemolytic. And the last one you have a 38. So that's 38,000. Is this guy UTI? No. Because McConkey agar you didn't see anything, and didn't meet the standards of 100,000 CFU per ml. So fine, survived, but go to see a doctor sometimes. Who else has a sample? Let's see what it looks like. Well, that looks good. Uh-oh. So let's see this patient. Nothing happens on the McConkey agar. That looks pretty good, not E. coli. Well, if you look at the blood agar, you actually see some alpha hemolytic there. But I doubt where this comes from. Maybe come from contamination. Because, surprisingly, this nutrient agar is nothing there. So that means the testing could be some sample from the bench or from somewhere. It's a contamination, cross-contamination. Okay? Because otherwise, you're gonna have some just pass it. So you have a 2 out of 3, which is we don't have anything. So that's okay. That's not bad. Who else have another plate? Yes, let's look at last plate. So this is from a male patient. Presumptive. Nothing grow on the McConkie agar. Very good. Basically nothing on blood agar. That is a cross-contamination, obviously. And you have only one cross-contamination, red colony, yellow colony on the neutral agar. So I would say this guy is good. Pretty healthy. Keep doing the good work. So that's what you do, and for your urine test, okay? And you can go ahead to record your notebook what happens, and for your 3 patients, you also can see the deep streak test, what it looks like. I believe it's negative. Is that right? It's negative. Then you're good, you survive. Don't worry too much. So this is our first urinary tract infection, like stimulated nurse and the patients. We talk about this. So today we're going to do a second one. Okay, we're going to do a second clinical testing. Will be throat culture. So how to do the throat culture? 2 of you gonna work together. We have a sterilized cotton swab right here. So 2 of you work together, get 1 cotton swab, and got 1 blood agar. Okay, that's the first thing. So we have a blood agar. We have a sterile cotton swab. Of course, this place you have to label. Bench number, name, and just write throat culture, TC. The date, 10/17, for example. Okay? So what's the job you're going to do? In our normal oral cavity or your tonsil area, there is a lot of normal bacteria there, you know that. And once the bacteria is translocated, if the condition changes, then it could cause a problem. Could become up respiratory tract. So this is what's going to happen. In this tonsil area will be a very good reservoir for normal bacteria. And today we want to test what kind of the bacteria it is. And I would love 2 of you gonna work together. 1 nurse, another one patient. So how are we going to do? One person going to open your mouth, another guy wear gloves, take 1 out of the cotton swab, and the person who is open the mouth pretend to be a patient and say ah, like that. And then the nurse will be using the cotton swab to touch the tonsil area. You need to scrub it, okay? Then get the bacteria. So, the cotton swab will be here. If you do not have tonsil, then walk around the bench. That's okay if 3 of you. If somebody doesn't have tonsil, If the whole bench doesn't have tonsils, then that's another story. You let me know, we'll rearrange it. Okay? Once you have this cotton swab has been, so let's say this cotton swab, you have been collected the culture, what we do? Go to the blood agar, very intensively streak the first cord. Okay, then you could use sterilized loop or you use metal loop, that's okay, and do a second cord. And then flame a little bit to a third one. Let's do third, okay? And this blood agar plates, we will use a candle jar to do the incubation. What is a candle jar? This is a jar. And we need to put a candle. So how we do? When you finished all these, your blood agar will move to here. Okay, once you move to here, what you're gonna do? I have this from Yankee Candle. So I'm gonna put the top. Does anybody has lighter or fire?
We have one on the top.
Oh yes, good. So we're gonna light it, and then we're gonna cover it. Now, what's the idea for this candle jar? Of course, we need another one. Okay. What's the idea for the candle jar? We need to create a micro aerophilic environment. And it is approximately 10 to 15% oxygen, 2 to 5% carbon dioxide, and left 70% nitrogen. Because this microaerophilic environment in the candle jar will favor beta hemolytic bacterial growth. So we want to see in you throat culture whether we can see some beta hemolytic bacteria. This is most likely is a pinpoint colony. And the detail is described on the chapter 19. And this is the second lab report working with your bench partner. So today you get a throat culture, we'll grow in the candle jar, and we will more to talk on Thursday. Thursday we will do a catalyst test to identify is there possible Staphylococcus aureus or Streptococcus, and then we also do the gram stem to see the morphology, gram reaction, what it looks like. I'll tell you, it's very interesting. Some of you find Staphylococcus aureus in your throat culture. Lots of you find Streptococcus, and some of you even find Corynebacterial diphtheria. So we'll talk about that on Thursday. Okay, it's a very interesting work, and you guys gonna do today is your throat culture. So it's very easy. We have four. Wait a second, not yet. 4 of here. So 4, 4, 8. That's 12. That is gonna be 16, 17. You have to wait a little bit. I only have like roughly around 13 here. I ask my students to grab more blood agar as soon as possible. Okay. So we'll do this. So I will ask one of the students to lighten the candle jar for the whole classroom. Another thing also important, when we do this candle jar, more or less we'll have some water. So we will put some of paper towel inside to absorb the water. Otherwise it's going to be very wet. And Thursday is exactly the time after 48 hours. 35 degrees Celsius, 48 hours, we will to see the pinpoint colony. Okay? Okay, some other work we're going to do today is biochemistry test. This will be a big deal for the final exam. So we have to talk about. This is biochemical test number 1. And usually, we have another fact sheet you could take it.
Okay. Yeah, I grabbed that.
Yeah. And the fact sheet is right here. on chapter 15. And most of you did very good for the exam 1. But I'm going to tell you, exam 2 will be difficult, a little bit difficult at least, because of this biochemistry test. Now, this is also in line with our lecture material, because our lecture we're going to talk about biochemistry testing also. Okay? So we want to talk about that. So first of all, overview why we can do biochemistry testing. Because there is a substrate, and the bacteria has enzyme. The enzyme can be break down. You just oh, that's good for the morning. Thank you. Thank you very much, Corey. Substrate have enzyme will generate products. Okay, these products, lots of the time, is gas, or some of the chemicals will cause color change. So we can recognize it. And sometimes will cause pH change. And we use pH indicator, we can recognize it. So this is something we could be partially identify a bacteria. So how do we know it is an enzyme? If you see anything ASE, it is an enzyme. For example, catalase, urease, oxidase. You see ASE here? That means it is an enzyme. Okay? But why I say it's like only 80% identification? Because lots of the bacteria could cause a very similar or same biochemistry reaction. Therefore, this is only give you some impression what a bacteria it is. Later on, most of the time, we need to use molecular methods to identify it. So, just want to let you know, we identify a bacteria, we cannot only rely on biochemistry test, but we have to do it because they'll give us lots of information. Okay, what we're going to do the testing today, we will do 2 tests. And there is a following about like another 5, 6, we'll talk later on. So today we will to do number 1, urea test. Okay, first of all, what is urea? Urea, NH2, CO, and NH2. This chemical is widely in the liver area existing. If bacteria has urease, it will break down urea, become ammonia and carbon dioxide. And ammonia is a weak alkaline, you know that. It is a weak alkaline, a pH is somehow about around 8. So how do we know the urea has been break down into ammonia and carbon dioxide? We have a pH indicator called phenol red. And this phenol red is very interesting. At pH 7, it is original color red. At pH 14, it is cherry red, even purple a little bit. And at pH 1 to 2, which is acid environment, it is yellow. Okay? So that's why we can test it. Now how we do it, it's very simple. You have 1 tube urea like this. And then use cotton swab to pick the bacteria. Can you please double check the bacteria? Is there incubation they have a bacteria?
Those are the
They're gonna be doing these, okay?
yeah.
So, what we're going to do the inoculation, we'll arrange. Because some of you will do E. coli, some of you will do bacillus, some of you will do lactobacillus, and some of you will do citrobacter. Okay, so we're going to do arrangement. everyone will only do one. So the work is very simple. This is urea solution. You're going to use a loop. Pick from slants, and then this urea solution incubate at 35 degrees Celsius 24 hours. We want to see the color change. So it's very simple, okay? That's a simple test. Okay, next test. This is kind of confusing. We have 3 tubes here. So we want to talk about it. Rayon, can you remind me what are these? What's the blue one? This could be phenol red. This is glucose. One of them are glucose, sucrose, and lactose. Which one? Which one is which? That you have to remind me.
Glucose is this one.
Which one is glucose?
This one.
That's glucose.
Yeah. This one is the dextrose glucose and this one is just normal glucose.
Okay, the dextrose glucose. How about that one?
This is the mannitol.
That's mannitol?
Yeah.
Okay. Okay, so the second test is 3-tube fermentation test. We will talk about the fermentation very detailed in the lecture. So what is 3-tube fermentation test? We have 3-tube right here. In today's class, it's a little bit different from the fact sheets because we don't have enough material. If you see the label right here, a yellow cup. Okay, you see a yellow cup. It is dextrose. And then you see a white cup. It is glucose. And the last one, when you see the blue cup, it is mannitol. Okay, blue one, it is mannitol. Now, usually what we do, we do glucose, fructose, or sucrose, and another one we do lactose, usually. But we don't have those, so we're gonna do dextrose, glucose, and the mannitol. Okay? So what's gonna happen? Fermentation, most of the time, is sugar. In the absence of oxygen, no oxygen, they will generate gas and acid. If those two products generate, we know it's fermentation positive. Now, how do we know? First of all, how do we know it is gas positive? If you look very carefully, inside the king cap tube has another small tube there. That is called the Duran tube. So, if you look very carefully, inside of these tubes, there is another tube inside called Duran tube. If the bacteria generate the gas, what happens? This Duran tube on the top will have bubble after incubation. So that means it's positive, okay? So you will see a very clear bubble on the top on Thursday. Then it's positive. How do you know it's acid? Same thing. If the solution turns yellow, because the phenol red is a pH indicator, it turns yellow, which means acid positive. Otherwise, it's negative. Or maybe alkaline material will generate. Okay? So today's work is also very simple. For the 3-tube fermentation test, everybody will get mannitol, glucose, and dextrose. Now there is a lab safety issue. Number 1 accident in the lab. King cap tube. Don't forget, inside another Duran tube. If this is broken, it's going to be horrible. Glasses everywhere. So make sure today only 4 tube for biochemistry test. You label them very well. Have your rack ready to get it. Okay? And once you get these, what's the first thing you do? Yes, very good. Don't forget, label. Okay, this is the key. I want you and your bench partner remind each other, did you label? Otherwise, on Thursday, you never gonna find your tubes. So remind your bench partner, did you label? What you gonna label? Bench number, initial, what are the bacteria you added there? And do not forget, you may be confusing about 3 different material there. So what do? You label ureas as a urea testing. Label dextrose, glucose, and mannitol. You could label D, G, and M to tell you what are the sugar are there. Okay, that's very important. Okay, next, let's assign the culture. From the beginning to the end, every bench. E. coli, Bacillus, Citrobacter, and the Lactobacillus. Lactobacillus might be Staphylococcus aureus. We don't know. We'll see. From here, E. coli, Bacillus, Lactobacillus, and Citrobacter. Here, E. coli, Bacillus, Lactobacillus, and Citrobacter. E. coli, Bacillus, Lactobacillus, and Citrobacter. Okay? You, 2 of you only responsible for 1 bacteria, and we'll share the results on Thursday. So that's the work we're going to do.